Okay, bio team. So you guys have learned that cells have some control over when and how they express their genes. That is, you've learned that they don't express all of their genes all of the time because they don't need to and they don't have the time or energy to do so. So one way that cells have of regulating gene expression is through something called inducible systems, which, as the name suggests, are systems that express genes only when they're needed, and they prevent gene expression when they're not needed. So one of the first discoveries of an inducible system is the Lac Operon, which is shown here below. And so the Lac Operon is a system by which bacteria can express the genes for digesting lactose. And so we can see these genes for digesting lactose down here. There's 3 of them. And we can also see in this Lac Operon a promoter sequence, which is the location where RNA polymerase can attach and then transcribe these genes. Now, a smart cell would only express these genes when lactose is available. That is, it only would express the genes for digesting lactose if there's lactose around to digest. And a smart cell would stop expressing these genes once the lactose runs out. So when there's no lactose around, there's no need to build the proteins for digesting lactose. So what this operon uses is something called a repressor protein, which attaches to an operator site between the RNA polymerase and the genes for digestion enzymes. And so this repressor protein blocks access to the genes until lactose becomes available. So we'll have some lactose enter the cell. Those are these little yellow guys here. When lactose enters the cell, it's able to bind to the repressor protein and change its shape. And that allows the repressor protein to release from the operator site. And when that happens, the RNA polymerase is now no longer blocked, and it can transcribe the genes for digestion enzymes. So the RNA polymerase will synthesize messenger RNA, and the messenger RNA will travel to the ribosome, where it's translated into the enzymes. And so once the enzymes for lactose digestion are made, the enzymes can start the process of digesting the lactose. So we'll watch that happen here. Now, once the enzymes have digested all of the lactose, including the lactose that had attached to the repressor is able to return back to its site on the operator, resetting this process. And so that's it. This is an inducible system in which the genes for lactose digestion are only expressed when lactose is present, and as soon as lactose is gone, they stop being expressed. At this point, you guys have a couple of practice problems. We'll see you next class.